Hi, welcome to a webinar about multiplex immunohistochemistry. This will be presented by my colleague, Dr. Craig Powell. He is Director of Technical Services at Vector Laboratories. We will discuss the selection of detection reagents and positive and negative controls to reduce cross-reactivity. At the end of the webinar, we will have a Q&A session. Please feel free to submit any questions to the GoToWebinar site. Now I present to you Dr. Craig Powell. Over to you.
Thanks, Byron. Hello, everyone. Good morning, good afternoon, and good evening, wherever you might be located around the globe. Thanks very much for tuning into our webinar. As Byron mentioned, today's topic is multiplex immunohistochemistry. Now, just before I launch into the webinar, I'd like to clarify the term multiplex. I see this word as being adopted by the life sciences to explain methodologies used to visualize and detect two or more targets in a given specimen. Now, multiplexing may involve fluorescence or indeed enzyme-based chromogens and look at genome profiling, protein expression, or indeed a combination of these on the same specimen. Now, these methodologies are gaining importance, as researchers investigate spatial relationships, intracellular signaling, or indeed the tumor microenvironment. Now, today, we're using the term multiplex to highlight multiple antigen labeling on the same tissue section using enzyme-based systems. So I'm giving a definition here of multiplex immunohistochemistry. Now, this is the same thing as calling it double labeling or indeed triple labeling if you have three labels on the same section. Now, on the right-hand side of this title slide, I have a great example here of double labeling. And now this is a section, this one over here, is a section of human breast carcinoma stained for a lymphatic endothelial cell marker. It's highlighted here in the purple. And the other marker is estrogen receptor, which is highlighted by the brown staining in these particular cells here. Now, you can imagine that if you just saw the purple stain as a single label by itself, you'd say, yeah, there are some lymphatic vessels that are present there. Or indeed, if you just looked at the brown staining, the estrogen receptor staining in those given cells, you'd say, yeah, there's estrogen receptor positive cells there. But when you put them together, it provides significantly more information and tells a much more compelling story. And it certainly looks like these estrogen receptor positive cells or cancer cells are metastasizing from a primary tumor through these lymphatic vessels. And so this image here is a great example. To me, it's the epitome of immunohistochemistry and being such a powerful technique to provide this type of insight in certain disease states. Now, certainly we hope that with the information we cover today, you'll be able to generate similar compelling data in your given field of research. So here's a quick overview of what we'll be looking at in today's webinar. So for those of you who have just joined us, just gone through a little bit of a term about multiplexing, highlighted the powerful application here of immunohistochemistry, and now we'll get into the webinar itself. We'll look at simplified double labeling, maybe for those who are new to the field or looking to introduce multiple antigen labeling or multiplexing, chromogenic multiplexing in your lab. And we'll look at considerations for multiple antigen labeling, and that'll take up the majority of the webinar. And then we'll sum up at the end and have a look at some tools and resources that are available. Okay, so some of you may have heard about double labeling. You may have heard that it's complicated and complex, difficult, a lot of optimization required. Well, what I've got here is what I'm referring to as a simplified methodology in order to obtain double labeling. And you do have to have a couple of criteria in place to use one of these given complete double staining kits. And the example I've got here is a sort of a flow diagram, a workflow, an abbreviated workflow of a double label methodology, whereby we have, say, one primary antibody that might be a mouse primary antibody binding to its target antigen. Here's a rabbit primary antibody binding to its target antigen. And then you'll detect them with their respective secondary antibody detection reagent. For example, this would be an anti-mouse HRP or peroxidase-based conjugate, and then here's your anti-rabbit with alkaline phosphatase, and then you develop those given signals independently with the appropriate contrast in enzyme substrates. So these are part of what we call our Impress duet double staining kits. And the caveats you need in order to do this easy double labeling would be using a human or indeed a non-human primate specimen, as in non-rodent. The two antibodies would have to be one raised from a mouse, one from a rabbit. You could mix those two together, and then you could apply this duet reagent, which is supplied pre-diluted, ready to use. They would bind to the respective primary antibodies, and then you develop those substrates independently. So this is double labeling in a single kit. All you do is add those primary antibodies. The other caveat would be just to ensure that those target antigens aren't completely overlapping at their site of localization. And that's a topic that we'll discuss a little more in depth at the end of the webinar here. But indeed, if you do meet those given criteria in your lab and you're new to the field, have a look at one of these kits. It'll get you up and running very quickly and reduce optimization time at your end. And just to give you a real life example of that, here's an investigator, Victoria Mulligan, had donated or submitted, I should say, this given image as one of our photo contest submissions in a previous year. And Victoria looked at two antigens, PCP4, highlighted by the red staining there, the vector red staining, and NUN, which is a common neuronal marker there. And this is in brain tissue. Obviously, human cerebellum is indicated there. So it just gives you an idea about a real life example of how one of these Impress duet double labeling kits could be utilized. So have a look at those, the clear demarcation there or contrast between those two antigens. See if that's something that might also be applicable in your given application. However, I'm certainly aware that biology is not as black and white as that, and there's a lot of gray areas. Indeed, different primary antibodies could be raised in a goat, rat, things like that. You know, and you don't get to decide what sort of specimen your tissue is from, and it could be rodent-based, for example. So we're going to go through scenarios then that if you already have in place a single label such as this, but you want to introduce a second or a third label, we'll look at some of these parameters. So indeed, this is another abbreviated workflow that you might already have in place. Again, let's just call this your primary antibody binding to a target antigen, then your secondary antibody, your polymer-based, it's called a peroxidase, and then you develop with a DAB substrate. And at this point, you're saying, okay, do I use the same detection system for a second label, or indeed do I use a different one? And what are the pros and cons of each of those? And this is where we'll get into some of these considerations for multiple antigen labeling. And I put together here a table broken down into what you should be determining at your end on the right-hand side of the table here, and indeed what to avoid when introducing multiple antigen labeling or multiplexing. Think of this as a little bit of a troubleshooting guide. Should something not particularly add up at your end, maybe you're getting some interference, have a look at some of these that we've indicated here in this table, work through them, and this should then help optimize the results at your end. So we're going to work through a lot of these in the webinar just now. But first off, in this top box here, I'd just like to point out that if you are going to introduce a second or a third label, make sure you're aware of the species of that primary antibody that you've got relative to the one you may already have in place and the species of tissue. And just ensure that that additional or that second or third primary antibody that you're going to be applying on the specimen does recognize the target antigen of that tissue species. And that's pretty crucial. Make sure it's been validated for immunohistochemistry, not Western blotting or something like that, but actual immunohistochemistry, and recognizes that species antigen. That's pretty crucial in number one step in trying to map out here the requirements for successful double and triple staining. Okay, so we'll look now at species cross-reactivity, and we'll work through our list right here. So imagine you might have a rabbit or a goat primary antibody. You're working on either mouse or rat tissue, and the second or indeed third antibody that you're looking to introduce here may be raised in a mouse. Just be aware of the fact that this actually could then be a situation where you're applying a mouse primary on mouse tissue sections, and that secondary antibody will fail to distinguish between the mouse primary antibody and anything that might be of a mouse immunoglobulin nature in the tissue, and therefore generate false staining or background in your given specimen. So I've got here as a bullet point a xenograft mouse model. What I mean by that is, let's just say you're growing up a human tumor in a mouse and you've been looking at it with a rabbit or a goat primary antibody. But if you're introducing a second label with a mouse primary antibody, that human tumor is actually vascularized by mouse blood vessels. There will be mouse IgG present in there. So when you apply an anti-mouse secondary antibody, you will probably generate background, false background or false negative staining due to that inherent mouse IgG. So just be aware of those given scenarios depending on the setup in your lab. Further down, I've got here about rodent on rodent, whereby perhaps you're applying a rat primary antibody onto mouse tissue, and there are degrees, percentages of cross-reactivity of anti-rat secondary antibodies with the like rodent species of mouse. So indeed, look for things such as anti-rat mouse adsorbed secondary antibodies that will help eliminate that particular cross-reactivity on mouse tissue. There may be other scenarios too. I've got hamster here, whereby there might be a trace amount of cross-reactivity of an anti-hamster antibody on mouse tissue. If that's the case, you could certainly apply a little bit of mouse serum to the dilution of that anti-hamster antibody. And I'm talking about a low percent of cross-reactivity, somewhere around, say, 2 to 5% cross-reactivity. You add in a little bit of mouse serum into that dilution, let it sit for about 30 to 60 minutes on the bench to absorb up any stickiness or cross-reactivity. Then you can apply it to the mouse tissue, and that will give you a much cleaner result. And the final bullet point here I've got about cross-reactivity is look at some of these ungulate species. I know goat primary antibodies are fairly popular out there. Anti-goat has significant cross-reactivity, 50 to 100%, with bovine IgG, sheep IgG. So if you have sheep or bovine serum, maybe a low-grade BSA is being introduced somewhere, either in a diluent, or if this is in a cell culture application, just be aware that an anti-goat secondary antibody may bind, probably will bind, to bovine IgG that may be introduced through a BSA or something like that. So just be aware of those given cross-reactivity parameters. All right, let's revisit quickly where we were with that Impress Duet kit. And we spoke about, for example, having a primary antibody from a mouse, the other one from a rabbit. Then we put on the goat anti-mouse peroxidase, goat anti-rabbit alkaline phosphatase, develop with your two substrates. Hey presto, you've got double labeling. And that's well and good. Just note that these two secondary antibodies are raised in the same species. Let's take a look at another scenario. You're going to use a mouse primary, a goat anti-mouse HRP, and then develop with DAB. But the second primary antibody you're going to introduce is GOAT, but you select the rabbit anti-GOAT alkaline phosphatase, and you're going to develop with vector red. That's well and good, but just be aware of the fact that there is this high potential for cross-reactivity between the goat anti-mouse and the rabbit anti-goat in this given scenario. And so what you'll have to do would be either substitute in, for example, a rabbit anti-mouse for that first label, which would require you to further optimize the dilution parameters for that. Or indeed, you could run the label sequentially, whereby you'd run the first mouse all the way through primary antibody detection reagent, all the way through to DAB staining. Then you'd repeat the staining, applying the second primary antibody, run through the rabbit anti-goat AP, and then develop with vector red. Obviously, you can't mix together the goat anti-mouse and the rabbit anti-goat. They're going to glom together, and you're not going to get any binding to your target primary antibodies. But you can run them sequentially using this chromogenic multiplex methodology, because these substrates do precipitate out as solids at that site of localization and almost put this protective shell over those first detection reagents and eliminate, by and large, cross-reactivity with these secondary detection reagents. All right, we're having a quick look now at the enzyme detection reagents and the substrate choices. I've got here the first bullet point on this very blank slide, sensitivity, and have a quick think about antigen expression levels on your given specimen for your second target antigen. And I'm here assuming that you have in place already a first label staining, but you just can't throw down a second primary antibody without due consideration of where that second antigen might be expressed, or indeed the level of expression. And knowing the degree of sensitivity there will help dictate the choice of detection reagents and the degree of sensitivity. Maybe you're using a one-step polymer base for an abundant antigen already. Maybe this second antigen has been upregulated. Maybe it's even of an unknown expression level. In that case, I would highly recommend going with a more sensitive detection system and a very sensitive substrate. And we'll cover some of that a little later on about the substrates. So just bear in mind the level of sensitivity that might be required when introducing a second label. Have a think also about some of the precipitate characteristics of these enzyme substrates. The peroxidase base substrates give a very heavy, dense precipitate at the site of localization, whereas the alkaline phosphatase are a little more transparent, a little more diffuse in the way they do precipitate. So certainly, if you were looking for intracellular markers, certainly the peroxidase-based substrates would be ideal for punctate sort of staining or very sharp, crisp membrane labeling that you're looking for. And then maybe the alkaline phosphatase could be used if you go back to the original, one of the original slides I showed earlier from Victoria Mulligan about the new N. Admittedly, that did use DAB, but more of an overall layering of that given target antigen, and that might help identify some of the underlying tissue architecture or morphology. So just have a think about some of those given characteristics. Also, if you have been struggling with, say, high levels of an endogenous enzyme, and more than likely that's going to be something like peroxidase, you could certainly consider swapping to 2 alkaline phosphatase-based detection reagents. And that way you'd sidestep quite easily the requirement of having to block persistent enzyme activity that certainly could give rise to false positive staining. Some other considerations would be the presence of inherent or endogenous pigmentation. And I've got here on this right-hand side endogenous melanin activity. It could be a melanoma, for example, whereby if you were to use the traditional peroxidase DAB staining, you wouldn't be able to differentiate the DAB from that brown melanin staining shown here. These deposits look very similar to that of DAB. So you'd have to go with a different colored substrate, such as we've indicated here, in this case looking at cyclin A. So the idea here would be to contrast against any inherent pigmentation that might be there, or indeed any of the other substrates you're going to use in that given application. You want clear, unequivocal demarcation of those target antigens. And have a look at the staining here we've got for hematoxylin, contrasting nicely against that, both of the target antigen stained with cyclin A, as well as the inherent melanin there. We'll talk a little bit about some counterstain choices a little later on in the webinar. Have a look at some of the solvent resistance of some of these particular substrates as well. And this depends a little bit on the intention of your given assay. Maybe you just want to do a quick stain and throw the slides out, and you're not particularly concerned about archiving them or indeed storing them for any length of time. In that case, you could go with a very quick aqueous-based mounting, and that way you don't have to go through what I refer to as more of a tedious methodology of doing the dehydration and clearing and then permanent mounting. Again, that depends a little bit on the scope of the project and whether or not archiving is important for you. And I've listed there also time and cost considerations. Mounting media, what's at your disposal? You know, mate, it is very easy, quick and convenient just to use an aqueous base media. You know, do you have to go out and purchase another permanent base mounting media? So have a think about these chromogen selections and some of these considerations when you're going to introduce a second or indeed third label. There are quite a number of different choices of substrate out there. And indeed, I've indicated here 3 different choices of an aqueous-based enzyme substrate. This is AEC. We've got it in 3 different flavors. Indicated here an economical version, the original one. It gives you a lot more working solution than some of these others, but some of these others are more sensitive. I'd say Impact AEC is probably about 2 to 3 times more sensitive than the original, and then the Impact AMEC Red is around 3 to 4-fold more sensitive than the original as well. So again, bear in mind sensitivity. If aqueous-based media is a way to go for you, and certainly convenience as well, maybe you could certainly have a look at either the Impact or the Impact AMEC Red. There are other choices too in terms of the more permanent HRP-based enzyme substrates. And we did look in that title slide at the combination of DAB and Impact, or VIP, I should say, the purple one. They give a very nice contrast, and there's a lot of different other choices you can put together. And here's for the alkalinephosphatase-based substrates as well. The more popular ones, the Vector Red and the Vector Blue. And the reason why I'm showing you all of these is you can use a lot of different combinations, color combinations for double and triple labeling on your given tissue specimen. So have a look at some of the tools and resources I'll show you a little later on here in the webinar. See what you think might look appropriate for your given application, and you can certainly apply those. But I'm going to say, you know what, that sort of muddies the water a little bit. Is not particularly helpful to investigators looking to get into the application. And so what I've got here is an easy way to help eliminate some of that guesswork on what color combination might work for you. More than likely, if you do have a single label already in place, you're probably using DAB or diaminobenzidine and a peroxidase-based methodology. That's great because DAB goes with pretty much everything. It goes with a lot of the other, or when I say goes with, it's compatible both in terms of its use and its color contrast with many of the other peroxidase-based substrates. This is the image from the title slide, DAB Vector VIP, DAB Vector SG. This is the smoky bluish gray color. Again, you can see a nice color demarcation there. Or indeed, you can put all 3 together on the one tissue section and get triple labeling. This is the VIP. This is a section of tonsil looking at some epithelium. Then you've got your B and T cells here highlighted with the DAB and the vector SG. And then DAB with nickel, this is the slate gray or grayish black color over here. You can combine that with the vector Nova red. And certainly DAB also plays nicely, or compatible, I'll use a more scientific phrase, with the alkaline phosphatase substrates as well. Here's DAB and vector red. These combine to be the two substrates included in that impressed duet kit. If that's not to your liking, certainly DAB can go or can be used, I should say, in parallel in combination with the vector blue, or indeed you can put those three together for a triple label. Vector blue, the heavier deposition here of DAB in these cell nuclei, or indeed vector red. So hopefully this gives you some ideas about how to get started in your given lab. Or maybe look at ways of improving the sensitivity in your lab too for multiple antigen labeling. This is a chart. I don't expect you to memorize this by any means. We've got this in one of our, in a couple of our guides, I think, our multiple antigen labeling guide, as well as in our IHC guide. Peroxidase-based substrates indicated here. A couple of different choices. For example, we've got the regular VIP and then the more sensitive impact. Same thing with DAB, the Nova Red, etc. And the alkaline phosphatase substrates as well. It gives you an idea about more economical versions and the higher sensitivity substrates, depending on, again, the needs at your end. All right, just to round out here our discussion on enzyme substrates, you know, the application here of immunohistochemistry or chromogenic immunohistochemistry is a little unusual in that there is inherent enzyme activity that you have to eliminate or get rid of, and then you're going to introduce that same enzyme, probably either peroxidase or alkaline phosphatase, as part of that detection system. So just ensure that the blocking is done appropriately, both for the endogenous enzyme activity, as well as potentially any cross-reactivity that you might see with, say, two peroxidase-based labels or two alkaline phosphatase-based labels, and make sure that those appropriate controls are in place. And just a little bit more on those a little later on here in the webinar. What is important too is the order of labeling, and this would depend a little bit on the antigen expression levels, which one is weakly expressed, which one is highly expressed, and then that would factor into the substrate choice. So this table here, which you may or may not be able to see, again, you can download this from our website, from one of the guides we've got. This would be the first substrate. Here's the alkaline phosphatase, and then your second substrate choice is over here, either another alkaline phosphatase or peroxidase. And then the minus signs would be showing an incompatibility, and the plus signs would be compatibility with the first or indeed second label choices. It's always an idea to label the weakest antigen first. When I mean the weakest antigen, the weakestly expressed antigen first, with the most sensitive detection system, and then follow that through with the more abundant antigen as your second label. That always seems to work best, at least in my given experience. All right, let's pivot now to controls. These are vitally important when you're introducing a second or a third label on your given tissue specimen. You have to use both positive and negative controls, and importantly, establish these for each of the target antigens that are corresponding to the detection system that you're going to use, whether it be peroxidase or alkaline phosphatase, and work these up as a single label on that target antigen, on that target specimen, I should say. Run them in parallel also as single labels, you know, to determine the appropriate staining patterns you're getting, you know, both for the positive control, yes, those cells are positive, and then compare that with your unknown or your test specimen, and have a look also at the absence of staining with the negative controls too. And these should also be incorporated in parallel when you do the staining in your multiple antigen labeling, because if anything untoward arises, maybe you see an absence of staining, you can always go back and refer back to these controls, positive and negative. And indeed, with a negative control, if you do get an unusual result, is that true? Is that accurate? Instead of scratching your head thinking, I'm not sure if that is accurate, you've got these controls in place to help give you confidence that, yes, what I'm seeing is a novel observation, and you can move forward with that given observation. Make sure you can repeat it, obviously several times, but nonetheless, it hopefully underscores the idea of why you didn't use and introduce these controls in your given system. Importantly, though, also a negative control, and I'll just read from the slide, helps identify any potential cross-reactivity between the labels, say first and second, for example, and any false positive staining that you might be reporting on. Obviously, you don't want to go off on a tangent on false positive staining. Again, this underscores the importance of using these negative controls. Let's have a quick look at some crucial negative controls here. So we've got here your first label in place, your first primary antibody, doesn't matter what it is, let's call it mouse, your second, your first secondary antibody, and then you're developing with your first substrate. So here's your first label, and then here's your second label, your second primary, second secondary detection reagent, and then second enzyme substrate. Let's call it, VIP. So you've got two peroxidase-based systems here, two different colors contrasting quite nicely. So the idea would be to, for a negative control, substitute in a non-immune IgG for this second primary antibody. And then you would continue to run this secondary detection reagent, apply that, and apply your second enzyme substrate. The idea being you want to ensure that this secondary detection reagent is not binding to superfluous primary antibody or indeed anything else in the detection reagent, and also ensure that this second enzyme substrate is not reacting to potentially residual or accessible enzyme, if it is the same enzyme-based detection system, from that first label. Indeed, if you do see any staining with this negative control where you're substituting in a non-immune IgG, eliminate this particular control, just apply substrate alone, and see if you see any cross-reactivity or staining. If you do, in that same sort of area of localization, obviously, you will have to consider putting in a step in here to quench some of that residual peroxidase activity. All right, hopefully that's clear for you in terms of the negative control. And certainly for a tertiary label, if you're going to run that, you'd have to run another series as well in the same manner. All right, so some of you might be applying, say, a two-step methodology or indeed an ABC kit for your double label. So here's your first label up here, first primary, first secondary detection reagent. This is your biotinylated secondary, avidin, streptavidin-based enzyme system, and then your first substrate, for example, DAP. And then you're going to run a second label using the same methodology. And what you'll have to do is substitute in, very similar to the one I just showed you, a non-immune IgG here, but then run all of these other secondary detection systems to ensure you are not getting cross-reactivity with any of the first label. Again, if you do see staining in this scenario, you're going to have to run further defined deletion controls, eliminating this particular step, eliminating that step, just applying substrate alone, and then work forward, applying the ABC enzyme reagent, and then the chromogen, then the secondary ABC and enzyme as well. So this will help give you confidence that if you are seeing novel observations, then therefore, if all of these check out, then that gives you something to report on, and hopefully great results and a paper coming out. All right, let's pivot a little bit now to counterstains. And in an image I showed earlier on with the inherent pigment, the melanin and the cyclin A expression, I did show the presence of a counterstain. It was blue hematoxylin. That was a single label. Now, what we're looking at here on the image on the right-hand side is a triple label. I did show this a little earlier. This was 2 alkaline phosphatase substrates, vector blue and vector red, in combination with this brown DAB. I think it's looking at Ki67 here. This, I think, was a cytokeratin, and I can't remember exactly what this is. Might have been CD34 or something. But nonetheless, on here, these are the 3 substrates. It's a triple label. And the question I posed here is, do you need another color? And what that really means is, do you need to introduce a counterstain in here and put a 4th color in place? And again, a further question is, does the counterstain add or indeed subtract or have a negative impact on your specific antigen staining? And on the example I've got here, I don't see that there would be a place for introducing a 4th color. Given the extent of staining you've got in the specimen, you know, all of the tissue is pretty much occupied. So putting in, you know, some contrasting stain, let's just call it a methyl green here, for example, you know, I don't think that that would help. I think it would potentially mask some of the staining and interfere with interpretation of your results. So indeed, you know, you have to ask the question about counterstains, and I've got the point here. You know, maybe you're new to the lab, you're following an established SOP. You know, try to question a little bit some of the methodologies in place. Indeed, I've got here, don't be a victim of lab dogma, whereby I've fielded inquiries, people saying, well, we've always done it this way. Well, that's well and good, but maybe once upon a time when that SOP was established, you were just doing single labeling, or indeed, you know, something with an inherent pigment like I showed before, with a single label and the hematoxylin, and that worked well. But maybe your staining has evolved, and indeed the specimen has evolved. You've changed specimens. You're looking at different target antigens, and now there's no real place to apply a counterstain. I'm just going to hammer this point a little bit because I feel this is very important. A lot of people just haphazardly throw a counterstain on, and you can really ruin a lot of hard work by doing that. And on this image here, I've got a very robust hematoxylin that is showing masking of the nuclear DAB staining you've got here. You want to be very careful in how you do apply the counterstain. Admittedly, this is a single label. I understand that, but just showcases how detrimental sometimes adding a counterstain can be to your given application. Now, I've got this from our friends at Roche, and they spoke about, you know, visual acuity of their given target staining. So just be very careful about applying it. Have a look at the target antigen staining, its localization. Is it also a nuclear-based antigen? And if you are going to be using, obviously, a nuclear counterstain, they're going to be overlapping. So make sure that you've got good demarcation between that, a very light counterstain. And certainly optimize the counterstain as well on separate tissue sections with some of the parameters I've indicated, time, pH, etc. What I've got here, though, is a very nice example of a double label with a hematoxylin counterstain. And I've got here, this was very nicely donated by our colleagues at Synaptic Systems. And you've got a DAB highlighted here, brown DAB, contrasting with the pinky red alkaline phosphatase, vector red here. And then the outer line, I should say, excuse me, hematoxylin counterstain, again, contrasting with these. And you can see a lot of different, you know, cellular architecture out here. And this is just a snapshot of the tissue, but you can certainly see other structures beyond this. This shows you how you can certainly appropriately apply a counterstain in a multiple label application. All right, let's close out here quickly with a bit of a chat about co-localization. And I do field inquiries about the use of chromogens, these chromogenic immunohistochemistry, of course, and the overlap of two antigens. And I've got here a nice figure that I took from this given paper down here in Nature Medicine. They actually used two antigens. One was BRDU, highlighted here with vector blue, and also nu-N staining with AEC. And they successfully co-localized these two target antigens using this methodology. And there are other examples out there, but this is a nice color plate I could show, at least educational plate I could show for this given webinar. I think that if you are going to do it, there's a couple of things you want to consider or crucial criteria for reliable, reproducible co-localization. One is the extent of the antigen expression and the overlap. And you can see here on the left-hand side, not every cell is positive for both of the antigens, and I call that 100% overlap. Sure, it's 100% overlap for the BRDU with the nu-N, but not vice versa. And I think that that helps with the interpretation of the staining. The other point would be substrate choice. And there is here two choices, one's peroxidase, one alkaline phosphatase. They do have different physical characteristics, kinetics. They do precipitate out in a different way, and it's referred to as the substantivity of it. And I referred to that a little earlier on about some of the precipitating characteristics of peroxidase versus alkaline phosphatase. And I think that these authors also had success with the order of the staining, whether or not they put the BRDU down first, followed by the nu-N. And certainly you can visit the given paper to clarify those points. But if you were looking at co-localization, you're going to have to give some serious consideration to some of these points I've highlighted here. All right, let's just sum up here quickly. And I've got a bit of a checklist here for successful chromogenic multiple antigen labeling or multiplex in the same tissue section. We did cover species cross-reactivity, both with the tissue and also the first or second label type cross-reactivity. Importantly, optimize each of the labels separately on different tissue sections. Work them up as single labels before you put them together. Also, one point I didn't really touch on was the use of a universal block or diluent. There are what I refer to as animal-free blocks that are available out there commercially or from other sources, maybe they're home brews. But if you're struggling with, you know, goat serum, rabbit serum, horse serum, whatever, donkey serum, whatever it might be, and do I have the right serum for the right detection reagent, give a bit of a think about using a universal block and diluent that you could use irrespective of what the primary antibody is or what the detection system is, whether it be peroxidase or alkaline phosphatase. And that way you wouldn't inadvertently use an incorrect serum, for example, and get false positive staining or indeed blocking of your detection. Certainly establish substrate compatibility. I did show you some of the charts earlier on, and the order of staining. And indeed, just because someone in their given application has showed that, you know, DAB and vector red has worked well, you don't have to follow that methodology. Look at some of the charts that we've got. Maybe again, this is looking at antigen expression and the extent of antigen that you might have in your prep. Maybe having the vector red first, followed by the DAB, might work better in your given scenario. Be aware, cognizant, of antigen co-localization, and have a think about, you know, whether or not both of those antigens are going to be co-localized in the same cell compartment of the same cells. If they're both two nuclear antigens and you're going to get 100% overlap, you may want to give pause about that given application. Also, a judicious use of counterstain, as I've indicated there, and you can certainly get trapped into masking your given target by introducing haphazardly introducing a counterstain. So just be aware of whether or not the counterstain will actually add anything of substance to that interpretation of your data, or potentially give you some false results. All right. At this point, I'd just like to indicate we do have some tools and resources available, our multiplexing guide and some other pamphlets and brochures available on our website. So I'd like to say thank you very much for your time. I sincerely hope this was informative, and I'll open the floor up to any questions you may have.
All right, thank you, Craig, for that great presentation. We'll proceed with the Q&A session. The first question is about the Impress Duet kit. Does it work with rodent samples? Why doesn't it work?
Okay, so the Duet kit actually has a combination of anti-mouse secondary antibody and anti-rabbit secondary antibody, your choice of either peroxidase or alkaline phosphatase on either of those. They're mixed in the same bottle as a pre-diluted, ready-to-use solution. And so my point being that they can't be separated. They're mixed together to make it a very easy methodology for double labeling. So the concern with applying the Duet kit on rodent tissue would be the potential of that anti-mouse secondary antibody cross-reacting either with, say, mouse IgG that might be inherent in the tissue, or rat IgG, another rodent or commonly used lab rodent species as well. And that would generate false positive results. So that's why I would hesitate recommending the use of the Impress Duet on rodent tissue. However, maybe the tissue you've got, and I'd just like to clarify, this would be, excised from a whole animal that was, at some point vascularized and would have immunoglobulin present. But if you know that there is no immunoglobulin present, maybe it's a cell culture or something like that, then sure, you can certainly apply the Impress Duet without concerns of that potential cross-reactivity. But on most tissue-based sections excised from tissue that was, vascularized, you just want to be aware of that potential for cross-reactivity.
Okay, great. The next question. What are the best color combinations for markers that are expressed in the same cell, co-localized, for example, CD3 and CD8?
Yeah, so that's always a little tricky. So those are a couple of membrane-based markers you've got there, and you might be looking at it in, general tissue, might be a tonsil or something like that. If you do expect 100% overlap with those membrane-based markers, let me just backtrack a little bit and say that when you do apply these precipitating enzyme substrates, whether it be 2-peroxidase, 2-alkaline phosphatase, or a combination of peroxidase and alkaline phosphatase, they precipitate out as solids at the site of localization. It's not like mixing wet paint where you're going to reliably generate a third color when they mix. They just don't operate like that. They've got unusual reaction kinetics and the way they do precipitate out. Indeed, some of them don't like precipitating on top of each other. And if one is already present, the other one might not precipitate there. So if you do face that scenario of CD3, CD8, what color combination would work well? I don't think I've got one that would work in that given scenario using the chromogenic methodology. You would probably have to go with a fluorescence-based system. What I will say, though, is if you do have two closely opposed or adjacent target antigens in the same cell, maybe one's membrane-based and one's nuclear-based or cytosolic, you could certainly use a combination of peroxidase or alkaline phosphatase-based substrates there. You know, again, as I pointed out, DAB is widely used. It gives a very nice, crisp demarcation. You could use that for the membrane marker in that instance, and then use an alkaline phosphatase for the nucleus, or indeed the cytosolic marker. But again, I'd hesitate to recommend co-localization of two membrane-based markers where you do expect 100% overlap.
Okay, thank you, Craig. The next question. I see that your images don't have nuclear staining. Would this be an issue for us to do data analysis?
So I did provide some examples of nuclear staining. Well, there's one for BRDU, and I'm trying to just, I'll go back here. So here was a triple label here for, hopefully you guys can see that. This is DAB triple label. This is the Ki67, a mitotic marker. So that particular image there does indicate nuclear staining for a given target antigen in a triple label. So certainly those were shown throughout the presentation. And indeed, the title slide also showed estrogen receptor expression in the nuclei of given breast carcinoma cells as well. So this is certainly something that can certainly be done in multiple antigen labeling, and that should not, you know, prevent imaging at all. Either this is going to be done through you know, ImageJ or spectral imaging or some other sort of software you've got. It's quite all right. So hopefully that clears up the, you know, potential miscommunication about not showing any nuclear staining.
Okay, great. The next question is about antigen retrieval. If antigen retrieval requires a different pH for each primary antibody, which one should I select?
Yeah, fair enough. So let's just say, for example, one primary antibody, it's recommended to use a citrate at pH 6. And if you're going to introduce a second detection reagent with a second primary antibody that has a recommendation of using an alkaline EDTA based or a tris based, let's say pH 8.5 or 9, which one do you use? Well, certainly I would apply the higher pH, at least from my experience, has been more of a universal unmasking reagent. So there's a couple of ways you can look at this. Number one, you can try both of those with the high pH. The EDTA seems to be a little more, again, as I said, just repeating myself, more of a universal unmasking salt than the citrate or the citric acid at a lower pH. Try that, see if it works for both antigens. If it doesn't, then certainly you could apply either one first. Try the citrate one with a heat-resistant substrate. So you'd work all the way from unmasking primary antibody all the way through to color development with something like a DAB, which is resistant to unmasking. Then you could go back, you could unmask with the EDTA, for example, or tris pH 9 unmasking if that target antigen requires that in the primary antibody, and then develop for that second color. So there's a couple of ways you could then target that. Try them with the higher pH, more of a universal marker. If that doesn't work, do them separately with a heat-resistant substrate, such as DAB is the first label, for example.
Okay, great. The next question is, what is the maximum number of enzyme substrates that I can use in multiplex IHC?
Fair enough. So we have shown in this webinar successful, reliable, reproducible localization of 3 colors. I think at one point we threw in a 4th as well. I would say that probably maximizes what you could do with chromogenic staining for again, reliable, reproducible, unequivocal staining. I think more than that, and you may start to run into some background problems with cross-reactivity as well. So I'd say certainly 3 or 4 would be a key target that you'd look for successful multiple antigen labeling using the chromogens.
Okay, great. The next question is it possible to use a primary antibody labeled with HRP or AP, like immunofluorescence?
Oh, absolutely, certainly, yes. Some investigators may have primary antibodies directly conjugated with an enzyme, as Byron mentioned there, either peroxidase-based or alkaline phosphatase. Sure, you could put on, say, you could mix those two together. If one is, say, a mouse HRP and a rabbit alkaline phosphatase primary antibody, you could even mix those two together and then react the substrates separately after that. One point of caution with directly conjugated primary antibodies is that you will experience low sensitivity with those. So if you're looking at a weakly expressed antigen or antigens of unknown expression, you're looking at upregulation or something like that, you may want to go with a more sensitive detection methodology. But nonetheless, yes, you can certainly use, say, a mouse primary antibody conjugated with HRP. And then if you've got a second primary antibody that's unconjugated, then you could use a different enzyme-based system or a different detection methodology to detect that, either with an impress polymer-based system or whatever you've got. So, yeah, certainly you can use that approach.
Okay, the next question is, what is the benefit of chromogenic multiplex IHC versus fluorescent NIHC?
Yeah, fair enough. I would say archiving would be pretty key. You can perform. Actually, I think that title slide, we've had that for years. You can go back and revisit that. Let's just say, this is an ongoing study. It's stretching over years or whatever it might be. You've got people coming and going in the lab. This is a research-based application, or indeed, in a CRO or wherever you might be. It doesn't matter, a biotech, a biopharma, and you want to go back and revisit that specimen, maybe different segments of that specimen. You want to take higher magnification images. Maybe you're finally getting around to getting that paper out. And you want to actually take pictures of that or better pictures of it now with some of the instrumentation you've got in your lab. You can do that with chromogenic multiplexing. You can't do that with immunofluorescence. It's gone the way of the dodo. It's faded out. It's lost its crispness. It's dissipated from the site of localization. Potentially after weeks or months, you're going to have to repeat that result, and that might be problematic for a lot of reasons. So I would say that would be one certainly significant advantage of using the chromogenic multiplexing over immunofluorescence. I would say also that the chromogenic methodology still gives you a great idea of some of the underlying tissue architecture and morphology. You can see, adjacent blood vessels or, whatever it might be, next to it, cartilage, bone, things like that. Other structures that might not be evident in immunofluorescence, and you might be only seeing half the picture with that, unless of course you did an adjacent H&E or whatever it might be. But then again, not everyone has access to some of the filters or a fluorescent microscopy or things like that in a given lab. Certainly, I think, as I've shown, you can easily get started with the chromogenic methodology. I think it's an extension of single label light microscopy. So there'd also be ease of application as well, whereas immunofluorescence might take a little longer to work up and optimize too. So I think there are some significant advantages in using multiplex. I'm not going to say that one is better than the other. It depends on your application, as I indicated too. With the CD3 and the CD8, it depends on the application and the scope of the project. Archiving might not be important to you, but nonetheless, hopefully that gives you some insight into some of the advantages of using the chromogen-based systems.
Okay, great. The next question is, can you comment on using 2 rabbit anti-mouse primary antibodies on mouse tissue? One antigen is nuclear and the other is membrane bound.
Yeah, certainly. So as I understand, primary antibodies raised in rabbit, you're applying them to mouse tissue, on adjacent or different, cellular structures, nuclear and membrane. So, yes, certainly you would run these sequentially. So you would detect the first primary antibody, first rabbit, with a given detection system. It could be peroxidase, you develop with DAB. Then you'd apply the second primary antibody and run that through either with the same enzyme-based detection system and use a different colored substrate, or you would use a different enzyme-based system and develop with a different alkaline phosphatase-based substrate. So that would have to be run sequentially to avoid potential cross-reactivity.
Okay, great. Unfortunately, we are out of time for this webinar. Thank you for attending this session. We will send you a recording of the webinar through email. And if we did not reach your question today, we will answer your question personally through email. Thank you for attending the webinar and have a great day.